Hi everyone, my name is Clericuzio and I'm a third year health science major and I'm currently on co-op in the Anderson lab at the Dana-Farber Cancer Institute. And I'm working underneath Dr. Anna Maria Gulla, who is an instructor in medicine within the Anderson lab at Dana-Farber, but she is also a group leader of her own lab at the Candiolo Cancer Institute in Candiolo, Italy. And so the project we are working on is investigating the role of GABARAP in autophagy and overcoming treatment resistance in multiple myeloma. So the potential impact of our project on the field of cancer is that our research could have a clinical impact as finding the correlation between GABARAP, autophagy, and being able to overcome the resistance from the current treatment that is in place for multiple myeloma could be translated into clinical medicine and can provide opportunities for physicians to treat certain multiple myeloma patients differently using a new combination of drugs that would be able to overcome drug resistance and hopefully enter into remission. And this would all be mediated by the expression of GABARAP within certain patients populations. So I have just been able to really find an immense appreciation for my research by knowing that there's a true potential to have clinical outcome. And I just feel like it has been really interesting for me to see the progression of this project from the beginning of my co-op to now. And so through this project, I have learned a lot of new skills. Um, One or two of the skills that I'd probably use on a daily basis is mammalian cell culture and Western blotting. So just a little overview, mammalian cell culture is growing cells and keeping them alive outside of their original tissue. And this is all doing it through sterile technique. And so using this sterile technique, I can do a lot of different in vitro studies, including different drug treatments and seeing how they affect the cells, phagocytosis studies, and also lentiviral infections, which would change the actual genetic makeup of the cell lines. And then I also do a lot of Western blot, which uses gel electrophoresis to separate out proteins within a sample. And this can really help see the up and down regulation through different samples, that whether they be a negative control and then treated with different drugs at different concentrations, or I can use this as a screening technique for different cell lines to see whether or not GABARAP or some other gene is expressed within the cells that we are studying. Also in this slide, you'll see flow cytometry and immunofluorescence, which are skills I'm currently learning. I do plan on staying on with the lab past the end of my co-op in June. And so these are going to be skills that I will be utilizing in another project that we're starting, but I'm really excited to just be able to grow my set of skills. And so flow cytometry, I will be doing a lot of extracellular staining of cells, which will be using flow cytometry to determine the expression of different genes within the cells that we are studying. And immunofluorescence will be staining cells with different antibodies to determine where actually in the cell that they are being expressed, and to see if there's any like co-localization of different antibodies together. But through my experience with the CaNCURE program, I have definitely been able to reaffirm my interest in pursuing medicine. I have been working within the field of cancer and just every day in the lab, and also hearing the speakers that the CaNCURE program brings in for us to learn from. I have really been able to grow my interest in medicine and honestly grow my interest within the field of cancer, because I never really saw myself going down into oncology. However, now that I've been working in it for 5 full months, I have really found an interest within the field of cancer and really within cancer research. And I could really now see myself continuing research as well. And I'd say one of the most valuable experiences that I've taken away from co-op has been the fact that I do see myself continuing research as part of my career. As a doctor, I do feel like I see myself in a clinical role as well now as a research role in addition to that. I kind of now feel like it's my responsibility that I'm going to be treating patients, and that it's my responsibility to not only be making sure that they're getting better, but also find new and novel ways to treat them better and maybe more efficiently, or be able to provide longer outcomes for their care. And then I think in the future, one of the things that I'm most excited for that I'm going to be able to directly translate what I've been learning here is being able to use my knowledge to inspire others. This fall, going to start volunteering with Med Science, which is a program at Harvard Medical School that takes high schoolers and teaches them the basics of medicine and science and how there's many crossroads between them. And I'm going to be able to directly use the knowledge that I've learned throughout this co-op experience to teach high schoolers these skills. And I'm hoping that I will be able to spark this sort of joy and this inspiration to go down into the field of research and medicine, the same way that the CaNCURE program has really sparked that interest within me.